Hi everyone, my name is Batuhan Çalık and I'm going to talk about the facts about facts. Sorry about the wordplay. So, as you may be wondering about the outline, here it is. I'll start with a brief information about what the fluorescence activated cell sorter technique is. Then I'll be explaining working mechanism of it, and I'll be using some examples from exciting articles. And finally, I'll be evaluating on its future use. If we go back at the late 1960s, scientists were trying to find a mechanism and invent an instrument to sort the differentiated versions of a cell or indicate and classify different types of cells. And in Stanford University, the scientists Bonner, Sweet, Hewlett, and Herzenberg and the other ones invented FACS to do flow cytometry and cell sorting of viable cells at the same time. This is the different part of FACS from the flow cytometry because it includes the cell sorting. And also, flow cytometry means the cell measurement of the flowing liquid. So they started with 1 to 2 cellular parameters, but over the years they have increased the number of measured FACS and their dimensions, and they are called parameters. And the speed of sorting has increased too. And their parameters have increased up to 32 different colors, plus 2 scatter parameters. And the working mechanism is mostly the same with its first invented form and today's version, but the components it's getting higher as it gets more complex. And the basic principle of flow cytometry is to measure single cells in a flowing stream of fluid. The cells pass sequentially through the beam, and fluorescent light from the cells gives rise to electrical signals. The stream is broken into a series of uniform size drops downstream of the laser, and the cell signals are used to give appropriate electrostatic charges to drops containing the cells. And the drops then pass between two charged plates and are deflected to appropriate containers. A beam of light, usually a laser, is directed at the stream of cells, and detectors are aimed where the stream of cells pass through the light. One lines up with the light beam, and it's called forward scatter or FSC, and some are perpendicular to the cells, side scatter, and it's abbreviated as SSC, and while other detectors are measuring fluorescence of fluorescent. The fluorescent chemicals that may be inside the cells or attached to the surface of the cells of their envelopes, and they can be excited to emit light, which is captured by the detectors in our mechanism. And there is a simpler version of the previous slide because it's from the first paper of the invention, and this is from another article, and it simply shows, let me show you, the uh, fluid cells coming from here, and we are using one or a couple of lasers, and the detector detects the light coming, and it shows the separation and the different cell types on a plot, and we can observe the results from there. And also here is there are two charged plates in here, and with the chemicals that we use, if they are electrically positive or negative, they are being filled in different cups, and then they are being collected. So um, this is the combination of light scatter and fluorescence signals that gives us information about the cells. I mean the side scatter and forward scatter. And these are again simple examples to understand the results of it. Here there are different three types of cells that accumulated in different areas, as you can see. And also in here it shows the when we use two different colors to indicate the cells, and if they are in different areas they look like these two colors, I mean the orange and green, but if they are together it becomes darker. And then I want to talk about the articles. FACS allows us to simultaneously stain, analyze, and then sort cells from small samples of human blood cells. And they can be from AIDS patients, infants, stem cell transplant patients, and others. And also, um, green fluorescent protein is another example of fluorescent protein that is widely used in FACS studies. It's abbreviated as GFP, and GFP is made by the jellyfish Acaria victoria and is spontaneously fluorescent. The gene coding for GFP has been cloned and is now widely used as a reporter gene because the extent of its expression is readily measured by FACS. Also, another article that I found interesting, using the Fluorescence Activated Cell Sorter, which is FACS, they have sorted leukocytes from maternal blood and obtained samples enriched for cells that reacted with an antiserum specific for paternal antigens. And they potentially carried by the fetus, but not the mother, and the enriched samples were then scored for cells containing Y chromatin, which could be derived only from a male fetus. The use of the enrichment technique facilitates detection of rare fetal cells in the maternal blood sample. More important, however, it provides an independent immunogenetic criterion for confirming the fetal origin of these cells. For example, if Y chromatin containing cells were observed among those sorted from maternal blood, cells of the newborn from that pregnancy should carry the paternal antigens detected by the antiserum. Thus, by examining FACS enriched samples and correlating fetal cell detection with newborn reactivity with the sorting antiserum, they have identified fetal cells in maternal circulation as early as the 15th week of gestation. I'm sorry, I had to check my notes because there are so many details. And then in this article, the contamination of testicular cell suspensions in cancer patients was held by two different approaches, and one of them was FACS. FACS analysis, it's the left one in here. The FACS analysis of a pure population of EL4 cells ended up with a high percentage of purity at the end, and this also shows that FACS can be used to ensure the decontamination of a sample of cells. The other technique ends up with distinct gates areas, and as you can see, they are R1 and R2, and uh, they follow the R2 gate with high percentage of purity in cells because we have to examine those cells. And also, as a different type of article, I want to show this one because I think they can influence someone to do further research about it, and it can maybe become their main interest. So, even though FACS based approaches in microalgal biotechnology have been applied in recent years. The current use of this technology has mainly been limited to the search for lipid-rich microalgae. Therefore, in order to fully exploit the capabilities of this technology, future work needs to focus on the establishment of FACS methodologies, and they are currently being used. But as I said, maybe one of you can, can do a further research, and maybe it can become their own interest. And as the first part of my presentation, I want to talk about the future use of FACS. I made a list of possibilities in technical and scientific uses of FACS, which are improving detection capabilities, speeding up hardware and automation, or clinical applications such as pharmacodynamic monitoring or disease activation state correlation, and also multi-dimensional assessments of cell signaling networks can be further researched. And I think there are more to observe and explore and improve about the fluorescent cells and the FACS mechanism. I hope we can learn more about them. And thank you for listening to me. I hope you get the information that I try to give to you. And if you're interested, here are my references. Take care. Thank you.